I welcome everyone to IMA Biotech's webinar today. Many thanks for joining us. Hope you all are safe and good. Just to introduce myself, I'm Pavitra, part of the innovation team associated with the PK and PD studies at IMA Biotech. I'll be the host for this webinar today. During the presentation, you'll be hearing from Dr. Corinne Ramos, Director of Innovation at IMA Biotech. But before giving her the floor, a quick reminder that you can ask your questions in the Q&A box at the bottom of the screen anytime during the presentation. We will dedicate time at the end of the presentation to answer all your questions. This webinar is about the growing application of spatial multi-omics for the identification of biomarkers opens new doors in cancer drug development. Just a brief intro about Dr. Corinne Ramos. Dr. Ramos joined IMA Biotech in the year 2020 as the Director of Innovation. With more than 20 years of experience in leading innovative research and clinical programs, Dr. Ramos has extensively worked on the identification and development of biomarkers to predict therapeutic outcomes. She holds a PhD in molecular biology from the University Paul Sabatier in Toulouse, France, and an MBA from Johns Hopkins University Carey Business School in Washington, D.C., USA. Dr. Ramos, the floor is yours.
Thank you very much, Pavi, for the nice introduction. And thank you very much for joining me today. I'm going to talk about, as Pavi said, the growing application of spatial multi-omics for the identification of biomarkers that could open new doors in cancer drug development. At the very beginning, I just want to briefly give the mission of IMA Biotech. IMA Biotech, we support key decision in drug development with next generation bioanalytical services that combine robust GLP and innovative spatial bioanalysis. Just a brief summary of the company and where we are right now. We offer drug discovery and development bioanalytical solution. Our services include pharmacokinetic, pharmacodynamic from plasma to single cell level, paving the way to advance the next generation of drug development and accelerate therapeutic decision. The company is globally located with 4 different sites. I'm in Lille right now. It's where we are performing the spatial biology as well as in Boston. The Salt Lake City facility has a strong expertise in RNA therapeutics in regular bioanalysis, and the Colorado Spring facility has very nice experience and high experience in regular bioanalysis for small molecule. We have more than 20 years of experience in PK/PD, and we are qualified as well for clinical investigation. Why? Today I'm going to talk only about the spatialomics. So the question is why spatialomics? It's really important. So tissue do function because cells are intricately spatially organized. And these cells are constantly communicating with each other by excreting different molecules and detecting the signal secreted from other cells as well as from the broader environment. If you really want to understand the mechanism that give rise to various disease states and identify therapeutic pathway for new treatment, it's very important to understand where each individual cell reside within the functional tissue and analyze the transcriptome while preserving cells in their morphological context to better understand the function. That's what at Atima Biotech, we are using spatialomics to combine the molecular scrutiny with spatial data on the cell localization in the interior of the tissue. To give we have at Atima Biotech a unique multimodal spatial biomarker platform. So those are the platforms that we are using. We have imaging mass spectrometry, where we can analyze over 500 molecules, imaging mass spectrometry from Bruker. We perform gene analysis on microdissected tissue, and we can profile up to 800 genes using the nCounter from NanoString. We are performing in situ hybridization, or could be standard immunofluorescence as well. And you have a nice representation on the right of in situ hybridization for the probe CD4, FOXP3, as well as the interferon gamma, as well as standard immunofluorescence performed for the CD8 antibody. We perform in this in low plex. 4-plex, it's our maximum here. We have as well a special transcriptomic platform where we can profile up to 18,000 genes using the GeoMX nanostring platform. And finally, we also have the imaging mass spectrometry technology where we can profile up to 40 markers on a single cell section using the Hyperion from Fluidigm. All this data, if you really want to interrogate the data, you need to have digital pathology analysis that is performed to really understand and integrate those data, as well as bioinformatics. And this is the services that we are providing as well. So what is the value of the spatialomics? The high multiplex spatial profiling increase the accuracy of the drug development with a deep profiling of the tissue heterogeneity and complexity. We use the spatialomics in different phases of the drug development. In preclinical, with biomarker and new target discovery for characterization of the tissue microenvironment in terms of disease characterization, or to look at the pathway modulation. In clinical research, this spatial omics are used in patient stratification to try to identify the biomarker that could correspond to the response of a specific therapy. Are used as well for efficacy biomarker and drug safety and toxicity. We applied the spatial omics to different therapeutic areas that include the immuno-oncology area, oncology, inflammatory, and autoimmune disease. But let's start with the example. So this is an example of the spatially resolved biomarker discovery. In that example, we have analyzed 2 frozen resected colorectal cancer patients. Those patients were at different stages of disease progression. We had one patient that has a pathology stage at very early, T1, and the other patient was a late stage at T3. So we did microsect in each patient, in each tissue sample that we got from the patient, just the tumor. And we extract RNA from the tumor contents. Once we have extracted the RNA, depending on the quantity of RNA that we are isolating, an amplification methodology is needed. And here that was the case for this particular example. And we did use this RNA that we amplify to profile what it's called the pan-cancer progression panel from nanostring that has over 800 genes that are involved in angiogenesis, in EMT, metastasis, and ECM, as you can see above. And those are the results. So we find that there was a tumor-specific biomarker that were upregulated in the late stage cancer. So the plasminogen activator inhibitor P1, PA1, or the SERPIN1, the 2 of them were upregulated in the late stage versus the early stage tumor. So those markers, they definitely contribute to the poor prognosis of patients with colorectal cancer. So this type of methodology allows us to completely uncover mechanisms that allow tumor to invite into the microenvironment, but as well discover potential biomarker as potential drug target. And we know for a fact that the plasminogen activator inhibitor 1, it's already been used as a potential drug target for certain cancer. The next example that I'm going to give you, it's to show you the value of the spatialomics. It's a multimodal spatial biomarker discovery. So this is an application where we could start investigating what would be the multimodality spatial signature that could be involved in either response prediction of a specific therapy or in discovery of biomarker. the way we worked out this particular example, that we did some quantitative mass spectrometry imaging on one section, and we did perform the GeoMX DXP. I'm going to go into the detail on the serial section. So the QMSE and the molecular content quantification will help us drive as well the protein profiling that we want to target specifically on the tissue. So here we start by analyzing 5 frozen resected, again, colorectal cancer. And we had 2 cancer at the stage 1, 1 cancer at the stage 2, and 2 cancer at the stage 3. And we used for the GeoMX DSP, we used the immune cell profiling and immune activation panel. And something, like of importance as well, even though we had quite a small, cohort here, we did look at 6 different regions of interest, which were pertinent to the molecular accumulation per sample. So you can see a workflow of this particular experiment where the QMSE, and in that particular example that I'm going to give, we were looking at 2 specific metabolites. So we target the quantification of the small molecule here in the term that we were looking at the lactate and the pyruvate metabolites, and looking at this heterogeneity of accumulation in those tissues. And once we had isolated the phenotype of accumulation of this metabolite, we overlay the QMSE image to our GeoMX DSP, and we draw the region of interest in order to deeply profile the protein content into each region of interest isolated. So this is how the mass spec imaging looks like. So you have an example of 2 tissue sections, one which corresponds to the patient at T1, that the other one that corresponds to a patient at T2. And we can look at the metabolite at the bottom of the slide, and you can look at the lactate and the pyruvate. What is interesting is you already start seeing some heterogeneity depending on the histological region between those 2 metabolites that have, already well known effect on immune suppression, for example. So they are very of particular interest. In the 2 tumor, in those tissue section, we can start isolating and identifying 2 types of metabolic phenotype. There is one which is high pyruvate, low lactate, and the other one which is low pyruvate, high lactate. And we're going to use this accumulation of metabolite and the identification of this phenotype in order to drive the protein profiling in those particular areas. So here, how does that work? So you have the molecular image on the top. You have the GeoMX image, which is part of, before getting into the deep profiling, there is a scanning part that could be used to identify region of interest using fluorescently labeled antibody, which is the case here. We had stained the epithelium content with keratin and the stroma with the marker CD45. So you can see a 9 green staining that corresponds to the tumor, and the stroma, it's in magenta here. So once we have isolated, and I'm not sure that you can see that, the phenotype of the metabolite, we overlay the metabolite phenotype onto the tissue section that are going to use for deep protein profiling with the GeoMX. And what we can look at, that there was already a differential expression of different markers according to the different metabolite phenotype isolated. So for example, for the one that's high pyruvate and low lactate phenotype ROA, we started seeing phenotype that are more related to the CD27, the CD8, to the activation of the cell. In the one that has low pyruvate, high lactate ROA, we start seeing some increase of expression of markers that are more linked to immune suppression. There was not probably enough sample for this experiment, so there were very few markers that were statistically different from this experience. But you can see that the ICOS marker was really high in the low pyruvate highlighted phenotype, while the CD127 was low in that regard. So we could isolate with this combination of metabolite accumulation, especially, and as well as we could isolate hot phenotype versus cold phenotype with the potentiality of finding new therapy drug to turn a cold phenotype into a hot phenotype. So I'm going to give you more example about the disease characterization or the response prediction using our platforms. So here we're looking at 2 fresh frozen human non-small cell lung cancer sample, where those are baseline, so they are naive treated sample, but they've been treated after resection with anti-PD-1 therapy. And we know what are the objective response of those samples. So we know that one of them has a complete response after the treatment with anti-PD-1 therapy, and the other one progressed onto therapy. We did look at the spatial biomarker using, and spatial transcript in that case, using the immune pathway RNA panel for nanostring. And again, we had approximately 6 regions of interest per slide. And you can see the representation of one region of interest that has been identified with using the PanSECA again to label the tumor, as well as the CD45 to label the stroma content. So here you can see right away that you have some molecular heterogeneity depending on either the complete response or the progression disease marker. And you can see that, for example, you have the drug target, first of all, is it's much more represented. So the drug target is PD-1. It's much more represented in the one that responded to the therapy. But you start seeing as well more markers that are linked to the T-cell activation, as well as the toxicity in the complete response. And I just represented on the right. Some of the markers to show how significant they were in being differentially expressed depending on the progression disease on the complete response. So here we were looking more at the targetable pathway in the non-responder. So the same experiment was performed with the same panel, but we were focusing specifically on the checkpoint inhibitor. And what you can see that there is an increase of the marker of those checkpoint inhibitor in the progression disease. So if you look at the ratio of those checkpoint inhibitor, and this is what you have on the right, to the CD8 cells, for example, you can see that there is a very significant difference between the progression disease and the CR, with the progression disease having much more checkpoint inhibitor, which was not taken into consideration in the therapy of this patient because it just received monotherapy. Anti-PD-1 and not anti-CTLA4, but it was much more expressed in this patient that progressed actually on the monotherapy. The TIGIT pathway was the same story. You can see that for the ratio of the TIGIT to the CD8, you have an increase of this ratio in the progression disease patient. So this experiment helped us to identify marker of resistance to monotherapy in the progression disease patient. And we can look, and I'm going to show you more some single cell experiment that to show that the co-inhibitor receptor on CD8 cells are very important here. So here, the way we are analyzing the result from not the single cell spatial transcriptomic, because as you have probably identified, the spatial transcriptomic from geomics, DSP geomics is not a single cell transcriptomic analysis. So to phenotype the cell and to really position the expression of every transcript in each very specific cell population, we are using a cell deconvolution we are using cell deconvolution with an algorithm developed by NanoString, which is called the spatial decon. So what you have on the right, it's a cell profile matrix output. This is a matrix that specifies the expected expression profile of each transcript on each cell type. And what you can see, for example, that the TIGIT are very much expressed in the Tregs, for example, or the TCD8 cells. So in that way, you can reposition in each transcript according to a very specific cell type. So here, the result of the cell phenotyping, and you can see right away that you have a huge heterogeneity from the graph on the left between the complete response and the PD marker. Each bar corresponds to a region of interest. And what you have on the right, it's a floret graph that represents directly onto the tissue, what is the amount of different phenotype that we just identified through this spatial transcriptomic. So you can see that in the complete response patient, as you expect, you have more CD8 T cells, which the phenotype has been just described, that you don't see in the progression disease. So there is a very nice heterogeneity of cell phenotyping that could be associated with the therapy response. Finally, a last example that I'm going to show you today regarding the spatial omics and the spatial biomarker. It's the methodology that could be used for patient stratification. So again, you're talking about resected, you're looking at resected FFPE lung cancer tissue treated again with an anti-PD-1. And you know that you have one patient that responded to this therapy and the other one that did not responded. Here we are looking at 18 markers that were imaged with IMC, with imaging mass cytometry technology. Those 18 markers are represented on the right, and you can see that they are they've been attributed, to very specific phenotypes. So you have marker for endothelial cells, marker of epithelial cell, generic T lymphocyte, but as well the T cytotoxic lymphocyte or the Tregs. Here is an image of this particular sample that went through the imaging mass cytometry with the staining of 18 marker. There is just a representation of 5 markers here, including the nuclei, because it's very difficult to visualize at once, like all the markers. So the FOXP3 are in red, the CD4 is in green, CD8A is in blue, and the pancreatin that represent the tumor cells are in yellow. So what we're doing once we have stained those tissue, we are detecting the nuclei. So you can see the progression disease patient on the left and the complete response on the right. And using our in-house algorithm, we can perfectly segment the different tissue section at very good precision and accuracy. Here we have a precision and accuracy that sits around 90%. From that, we can do the cell expansion that allow us actually, to start building the cytoplasm around, the nuclei and have a good representation of the total volume of the cells. So again, you're looking at the progression disease on the left and at the complete response on the right. After that, we are performing classification. So at the very beginning here, it's just a determination of the cell type abundance in the region of interest. So we're looking just at the non-immune cells, T immune cells versus the T cells. So here you can see right away that there is definitely a different cell abundance difference between the progression disease patient and the complete response patient. If you go a little bit more into detail in phenotyping, you can see that there is a huge difference in terms of the TMA composition and complexity, actually, of the cell composition between the progression disease and the complete response, with fibroblasts being approximately at the same level between those two patients or the two sections. But you can tell right away that you have T cells, which is much more present in the progression disease patient. You have on the right an abundance of the CD8 positive cells as well, the PD-1 positive cell. And you can see that there is a difference in percentage between the complete response and the PD. So you can tell from this experiment that the immune cell infiltration, there is definitely an immune cell infiltration into the microenvironment of the complete response patient. And the percentage of the cell within the drug target, it's for the complete response patient, it's over 10%. Looking a little bit further into looking a little bit further into the phenotyping and more into the cell-to-cell interaction, we are performing two different experiment of spatial analysis. One on the top, which is looking at the cell interaction. So we can just look at the very specific cell population. Here we are looking at the T cytotoxic and its relationship and their relationship to the epithelial cell, which are the cancer cell, and the B cell. And you can tell not only that there is a very high, proportion of the T cytotoxic in the complete response patient, but they are also, very close to the epithelial cell. The last experiment that we perform is to look at the spatial proximity analysis. So this is exactly what you're looking at the bottom of the screen, where you can see all the different macrophage, all the different cell types, which are represented on the list on the right. And the red circle represent the T cytotoxic cells. So you can see, first of all, there is again, a much more volume of T cytotoxic cells within the environment of the patient that completely responded to the therapy. But this technology is very much used as well to identify some very specific hub of different phenotype that could be predictive of new potentially therapeutic target or predictive or have a much better accuracy for prediction of the therapy. So I'm going to stop here, and I would like, first of all, to thank you for your time. So if you have any question, you can contact either Erica for US or Florian for Europe. So thank you very much. And I'm going to take some question, I guess.
Thank you very much, Dr. Ramos. So let's take some questions. So before that, everyone, please free to ask your questions in the Q&A box located at the bottom of the screen. And I already have some questions that came in for you, Dr. Ramos. So can we customize the proteins and gene panels or the other panels with these technologies?
We can. With the imaging mass cytometry, you can definitely customize the antigen of interest or the one that you're interested in. We have already, a huge experience and have validated quite a lot of marker relating to the immune portrait or immune cell type, but we keep, validated and customizing new antibody for this particular technology.
Okay, thank you. And another question, so can we use this for fresh frozen samples?
So all of the technology that I show you today, except, like for the quantitative mass spectrometry imaging, can be used with either frozen or FFPE tissue.
Okay, great. So, and can the technology integrate RNA probes?
So we're looking into, so obviously we are doing spatial transcriptomic, and we try to go back actually, like to the single cell evaluation by doing this deconvolution algorithm. We're using as well, like low plex RNA-scope. So but we are looking into developing high throughput RNA technology, transcriptomics, single cell transcriptomic.
Okay, great. So how much surface can be analyzed in one study?
How much what? Surface?
How much surface can be analyzed in one study?
So the IMC, it's not a very high throughput technology in that regard. I mean, you can definitely do, like the whole tissue section, but it will be very instrument intensive. So we usually do, we have a resolution of 1 millimeter square. For the GeoMX, the minimum region that we can do, the maximum region per region of interest that we can do, it's 600 by 600 micron. But we can do, like multiple region of interest, so we can cover better actually, like the surface of the tissue section with a technology like the GeoMX.
Okay.
I have a question as well that I'm seeing in the chat. What is the maximum size of the tumor section to measure the specific cell biomarker to identify cell phenotype within a tumor? So we're working with 4 to 5 micron in FFPE, and we do between 7 and 10 for frozen. Do you have experience? Yeah, go ahead.
Yeah, it's followed by another question too.
Yes, I see that there was, do you have experience with intratumoral efficacy assessment at the phenotype level? So this is exactly what we are doing in the Department of Innovation at IMA Biotech. So we're going to mix the efficacy with the cell phenotyping in order to have a completely portrait of the biomarker efficacy. What I can say already that we have either already, not maybe at the single level, but we have experience looking at efficacy biomarker, and we do have experience already looking at the phenotyping. This is just exactly what I just showed you. We're going to miss that experience at the cell level, at the single cell level, to make sure that we can do both at the same time.
And there's another question too. Can you correlate drug concentration with tumor efficacy using the combination of small molecule determination and cell phenotype identification, as well as apoptotics or cell death biomarkers?
So this is a very good question, because this is exactly what the workflow I show regarding the metabolite accumulation with that drive actually the spatial profiling in terms of protein. This is exactly what this workflow is for. So we can combine the drug accumulation using the mass spectrometry imaging with very specific transcriptomic and proteomic profiling in that region of interest, in that accumulation of the drug.
Cool. Another question. Do you also use GeoMX for protein profiling? Yeah. And what
yes, that was actually an example. Maybe, like I was not very clear. So I show you, like for the GeoMX, 2 different examples. So there was one on transcriptomic and the other one on proteomic. So we use both. Yes, we use both, and we can go up to 100 protein with the GeoMX. And we also have experience at customizing a new DSP antibody that are not within the panel, the control panel for NanoString.
Okay. Yes, and is it possible to segment any tissues and cell types regardless of the tumor types, the immune cells, etc.?
So we do have a flexibility in terms of segmentation. I guess we're talking about the digital pathology analysis here. We have completely customized and in-house and proprietary algorithms that help us to customize, to completely segment in a very pertinent way. And I guess, the question is around the complexity of the cell composition. We have this flexibility of being, very specific to immune cells from, other cell type, for example.
Okay, and another question, how did you co-register the QMIC data to the NanoString?
So we just overlay directly the image, the molecular imaging directly onto the GeoMX. This is very easy to do.
Okay, so we don't have any more questions. So it was very nice. Thank you very much, Dr. Corinne Ramos, for the insightful webinar. And thank you everyone for joining us today. So we come to the end. We'll be emailing you the replay of the webinar soon. Don't hesitate to reach out to us in case of any questions meanwhile. I wish you all a very nice day for those based in the United States, and a very nice evening for the ones in Europe. So stay tuned for our next webinar with another interesting topic soon. Thank you very much.
Thank you, Perry.